Welcome to my channel. I'm making this video on monosaccharides and I'm making this lecture from the Voet and Voet Biochemistry. Monosaccharides or simple sugars are aldehydes or ketone derivatives or straight chain polyhydroxyalcohols containing at least 3 carbon atoms. So monosaccharides are also called simple sugars and they have functional groups such as aldehydes or ketones, and they are at least 3 carbon atoms, and they are also called polyhydroxy alcohols because they have more than 2 hydroxyl groups in their structures. As you can see, in the D-glucose form we have 1, 2, 3, 4 and 5 hydroxyl groups, and in the D-ribulose we have 1, 2, 3 and 4 hydroxyl groups. That is why they are called polyhydroxy alcohols. And such substances, for example D-glucose and D-ribulose, cannot be hydrolyzed to form simpler saccharides. So they cannot be hydrolyzed into simpler uh, saccharide molecules. In this section, the structure of the monosaccharides and some of their biologically important derivatives are discussed. First of all, let's talk about the classification. Monosaccharides are classified according to the chemical nature of their carbonyl group and the number of their carbon atoms. If the carbonyl group is an aldehyde, as in glucose, the sugar is an aldose. And if the carbonyl group is a ketone, as in ribulose, the sugar is a ketose. In an aldehyde functional group, the carbonyl group is bonded to a hydrogen at one end and to a carbon atom or alkyl chain at the other end. But in a ketone is made up of a carbonyl group which is bonded to two other carbon atoms or alkyl chains on either sides. As you may already know that carbon can form 4 bonds. So in the aldehyde it forms 2 of its bond with oxygen, 1 of its bond with hydrogen. These are compulsory, and the fourth bond it can make that bond with a hydrogen as in formaldehyde, or it can make with another alkyl group which includes carbon molecules. So this the fourth bond of this carbon can vary. But in the case of ketones, the central carbon, which is the carbonyl carbon, is doubly bonded to an oxygen atom to form a carbonyl group, and the two other bonds of the carbon can only be made with carbon atoms only. So it cannot make bonds with any other atom. Another thing that you should note here is that the aldehyde functional groups are present at the terminals of a carbon chain. It is not internally present within a carbon chain, but the ketone group is only present within a carbon chain. It is not present at the terminals of a carbon chain. So if the carbonyl group is an aldehyde, as in glucose, the sugar is an aldose, and if the carbonyl group is a ketone, as in ribulose, the sugar is a ketose. The simplest monosaccharides, those in 3 carbon atoms, are trioses. So trioses are those monosaccharides which have 3 carbon molecules. Those with 4, 5, 6, 7, etc. carbon atoms are respectively tetroses, pentoses, hexoses, and heptoses. These terms may be combined so that, for example, glucose is an aldohexose, whereas ribulose is a ketopentose. So glucose has an aldehyde functional group, that is why it is called an aldose or aldo, and it has 6 carbon molecules, so it is a hexose. So we combine both of these terms and make it hexose. And ribulose has 5 carbon molecules and has a ketone functional group, so we call it as a ketopentose. Examination of D-glucose's molecular formula indicates that all but 2 of its 6 carbon atoms, such as carbon 1 and carbon 6, are chiral centers. So the carbon number 1 and carbon number 6 are achiral, or they are not asymmetric. They are symmetric because the substituents present on these 2 carbons are not, all the 4 substituents that are present on this carbon are not different. So a chiral molecule or a chiral carbon is that which have four substituents bounded to it, but all those 4 substituents are different. So that glucose is 1 of 2 to power 4 is equal to 16 stereoisomers, because the four chiral carbons of this glucose are going to bound the substituents in different 3D conformation, or different the stereochemistry will be different, due to which we get 16 stereoisomers of this glucose that comprise all possible aldohexose. In general, the n carbon aldoses have 2 to power n minus 2 stereoisomers, because the carbon number 1 and carbon number 6, as in the case of glucose, are achiral. So we will be left with only those carbon atoms which are chiral. The stereochemistry and names of the D-glucoses are presented in figure 11.1. So you can see that first of all we have a 3 carbon monosaccharide, which is a diglyceraldehyde. Then we have 4 carbon aldoses, and we call the 3 carbons are called aldotrioses. The 4 carbon are called aldotetroses. In aldotetroses we have d-erythrose and d-threose. In aldopentoses we have d-ribose, d-arabinose, d-xylose and d-lysose. And in d-hexose we have d-allose, d-altrose, d-glucose, d-mannose, d-glucose, d-idose, d-galactose and d-tallose. The most common aldohexoses and aldoses that occur in biological systems are boxed here, which are the D-glyceraldehyde, D-ribose, D-glucose, D-mannose, and D-galactose. These are the most common biological monosaccharides that occur in biochemistry. The Emil Fischer elucidated these configurations for the aldohexoses in 1896. According to the Fischer convention, these sugars have the same absolute configuration at the asymmetric center farthest removed from their carbonyl group as does D-glyceraldehyde. So in D-glyceraldehyde, as you can see that it is a 3 carbon molecule. The central carbon or the second carbon of this molecule is bonded to a hydroxyl group on its right, so that's why it is known as D-glyceraldehyde. D is indicating dextro, which means right, and if this hydroxyl group was on the left side of this molecule, we would call it as L-glyceraldehyde. L stands for levo, which means left. So this is a molecule, we can call it as, this is a standard molecule that we use for naming the D and L forms of the sugar. So if the conformation of the chiral center that is furthest away from the carbonyl group is similar to the central carbon of glyceraldehyde, we will call that sugar a D sugar. And if the hydroxyl group at that achiral center farthest away from the aldehyde functional group is like that a hydroxyl group is present on the left side of that molecule, we will call that molecule an L sugar. As you can see in the case of glucose, so the carbon number 5, this carbon is the asymmetric carbon that is farthest away from the aldehyde group. So the conformation of hydroxyl group on this carbon is that the hydroxyl is present at the right side of this carbon molecule, that, and this conformation is similar to the conformation of the central carbon of D-glyceraldehyde. That is why we call it as D-glucose. And if we reverse this conformation, if we reverse the conformation around this carbon, that in such a way that the hydroxyl group is present on the left side, we will call it an we will call it as L-glucose. So the L sugar, in accordance with this convention, are mirror images of their D counterparts as shown below in the Fischer projection of for glucose. Next we have sugars that differ only by the configuration about one carbon atom are known as epimers of one another. The D-glucose and D-mannose are epimers with respect to carbon 2, whereas D-glucose and D-galactose are epimers with respect to the carbon number 4. So if we look at the glucose and mannose, the structure of glucose and mannose, we will see that the structure of D-glucose and D-mannose is same, but the configuration around the carbon number 2 is different. In glucose, you can see that the hydroxyl group is present on the right side, and in D-mannose, the hydroxyl group on carbon 2 is present on the left side. So they differ only at configuration around a single carbon atom. So that's why they are called epimers of each other. Let's look at the other pair of D-galactose and D-glucose. In D-galactose and D-glucose, as you can see, the conformation of hydroxyl group on the carbon number 4 is differ on the carbon number 4 differs in this pair. In glucose, as you can see, the hydroxyl group is present on the right side of this carbon, and in the case of galactose, this hydroxyl group is present on the left side. So these are called C4 epimers, and glucose and mannose are called C2 epimers because the conformation of hydroxyl group on the carbon number 2 is opposite, and in the case of glucose and galactose they are called C4 epimers because the conformation of hydroxyl groups on the carbon number 4 differs. But mannose and galactose are not epimers of each other because they have two carbon molecules that differ in configuration. In mannose, the carbon number 2, and in galactose, the carbon number 2, the hydroxyl group conformation in both of these molecules is opposite, and also at the carbon number 4, the conformation differs. So that is why these are not epimers, because they differ at two carbon atoms. But epimers are those molecules which differ only at one carbon atom. Therefore, D-mannose and D-galactose are not epimers of each other because they differ in configuration about two of their carbon atoms. D-glucose is the only aldose that commonly occurs in nature as a monosaccharide. Okay, the D-glucose only is the only monosaccharide that is present as a monosaccharide in nature. Okay, it is the only aldose. However, it and several other monosaccharides, including D-glyceraldehyde, D-ribose, D-mannose, and D-galactose, all the other uh, monosaccharides, and to some extent D-glucose, they are part of they are important components of larger and complex biological molecules. L sugars are biologically much less abundant than D sugars. The position of their carbonyl group gives ketosis one less asymmetric center than their isomeric aldoses. So the ketone groups are present within a chain, so they will have isomers one less than the aldoses. If we have a glucose molecule, as we have seen that glucose has 16 stereoisomers, but if we talk about a fructose, which is also a 6 carbon molecule, it will have 15 stereoisomers. So the formula for calculating the stereoisomers of ketose, a stereoisomers with an n carbon ketosis therefore have 2 to power n minus 3 stereoisomers. Okay, those with their ketone functions at carbon 2 are the most common forms. So the ketone can be present at carbon 2, carbon 3, carbon 4, etc. But those ketones which have their ketone group at, or which have their carbonyl group at the carbon number 2, they are most common in the biological systems. Note that some of these ketoses are named by the insertion of ul before the suffix os in the name of the corresponding aldose. Thus d-xylulose is the ketose corresponding to the aldose d-xylose. So so the name of the aldose can be modified in such a way to make it a ketone. Dihydroxyacetone, D-fructose, D-ribulose, and D-xylulose are the biologically most prominent ketoses. As we can see in this diagram, that dihydroxyacetone is the simplest ketose with a with three carbons, and the carbonyl is at the carbon number 2. Then we have a 4 carbon ketose, which is a D-erythrulose. Then we have 5 carbon ketoses such as D-ribulose and D-xylulose, and then we have 6 carbon molecules: D-cyclose, D-fructose, D-sarbose, and D-tagatose. And the most abundant or the most common ketoses are boxed, which are D-fructose, D-xylulose, D-ribulose, and dihydroxyacetone.